I can actually have
Yeah, I didn't appreciate that.
because I'm not sure which way that goes.
7 to 9 p.m.
She
Yes, if you don't ever say that.
Kind of.
got that Gatorade.
Invite friends. Family.
All right, we'll get started. Let's start with what's on the exam. So, the format for this one is similar to the last in terms of the types of questions. So there are, if I'm not mistaken, there are 5. I think there are 6 multiple choice on this one. Roughly 10 to 20 fill in the blank type questions. I can't remember the exact number. And then there are 4 short answer, and by short answer I mean they could be either answered by phrase or essay style that could be probably answered in maximum 4 sentences or so, roughly. Okay. The in-class portion is 85 points. The last 15 are going to be the take-home part that I'll be sending out later today. And the format of this one is a little bit different from the last one in that this is a single case study. So it is a case study on one bacteria, and what that means is that answers to previous questions could help you out on future ones. Okay, so pay attention to that as you're going through it. The exception to that is the take-home portion. That has nothing to do with the rest of it. Yeah, or even like there's one part where there's one question where I say like based on the answers to the first 11 questions, you should be able to find evidence in here that supports your answer for this answer. So what's that evidence? Emily. No, actually it's not, that's not what I'm saying. But if you were to get the first 11 wrong, but yet somehow get the 12th one correct, then the way that I grade tests like this, and this is true on the last one too, I'm looking at consistency. So in other words, if it's a multi-part question and you get the first answer wrong, as long as you're consistent through the remaining pieces, I give you the credit for the rest of them. So it's not like an all or nothing thing. Okay. And I don't want to try to give this away, but basically, nope, I'm not going to. I'm trying to think of something analogous. So an example would be like, think back to the last exam. I'll use an example from the last exam in some ways. If I were, actually, no, I'll give you an example from this one. An example from this one would be like, you've got to identify the growth parameters of a microbe. That's one of the objectives for this one. So is it a mesophile? Is it a thermophile? There's going to be a question that says, based on the set of characteristics of this microbe, give me a couple of ways that you can inhibit its growth. So that's one where you have to use the previous answers to come up with some methodology. That's what I mean by that. And regardless of whether you're right or wrong on those previous ones, I will grade you based on how you, so like if you incorrectly identify it as, for example, a thermophile, as long as you've said like, I could chill it and that would stop the growth because it's a thermophile, thermophile's like really high heat. That's perfect. That still works as a response. Okay? Does that help out, Emily?
Yeah.
To clarify? Okay. Okay, so I will be here, or the goal is for me to be here 10 minutes beforehand so that when you guys get in here, you can get started right away. We'll space out every other row like last time as well so that I can make sure to answer questions when they come up. But let's go over the objectives real quick, and I wanted to just kind of give a couple of examples of what I mean by these so that there's no questions. So the first one there, identify the 4 stages of growth. Basically, be able to identify or describe lag, log, stationary, and death phase of microbial growth. For classifying, this one is, remember the 4 major classifications that we've used. So we've classified microbes based on their ability to grow over a range of pH values, or grow over a range of temperatures, over a range of salt concentrations, and then whether they are obligate aerobes or anaerobes based on where they grow in a 5-glycolate tube. In line with that, you should know how oxygen will affect organismal growth. So for example, we've talked about one major difference between obligate anaerobes and aerotolerant anaerobes. They both perform, or they both can perform fermentation and also perform anaerobic respiration, but what is one difference between them? Whether they have catalase or not. So trying to keep in mind, when would an organism be required to have catalase, or which ones would we expect to have catalase based on where they grow in a thioglycolate tube, is another characteristic that we group with that. 5.4, know the structure, or sorry, less about the structure, but more about the function of spores, when they're likely to form, who forms them, are important points there. And then for the 5th one, describe methods for measuring growth. Essentially, for this one, reminding yourself of having a basic understanding of how we perform spectrophotometry, a little bit behind the theory of it. So we said light gets shot through a sample, and we basically capture how much light makes it through. That gives us an idea of density. Bless you. And then also direct methods of count, serial dilution and plate count. Okay. And then calculating and evaluating growth. There is one problem that may require you to use a calculator on the exam. Definitely the take-home portion I will have you doing some calculations for. Okay. All right. Next set. So for the next set, describe physical and chemical controls. We talked about a variety of different methodologies. So we talked a lot about different heat methodologies, pasteurization, autoclaving, as mechanisms to kill organisms with high bursts of heat. We talked also about cold treatments, so refrigerating, freezing as well. Some other examples that would fit under this would be using acids, changing salt concentration, or drying out samples, and also radiation, either in the form of UV or ionizing radiation. We spent less time on the chemicals, and so I'm not asking you to memorize every single chemical that we talked about. Instead, what I'm asking you to be able to do is to define a chemical as a disinfectant, or an antiseptic, or a degerming agent based on the context of the problem. Okay, so being able to compare and contrast those, which is kind of 6.2 there, is really the important point from those chemical methods of inhibition. The third one, developing strategies. What I mean by that is, if an organism is a mesophile, how do you kill a mesophile? Heat it, or you could freeze it. That's what I mean by this one. So given again the organism and its basic characteristics, coming up with a logical strategy to inhibit its growth. And then 6.4 there kind of goes with 5.6, and really analyzing data to evaluate the effectiveness. That's the take-home problem. I mean, that's really the take-home problem for this exam. Doesn't mean that it won't show up somewhere else, but being able to analyze a graph or a table of data and then determining whether the method is effective or not, and describing that will be important. This also includes broth dilution assays and disc diffusion assays, so knowing how to interpret those as well. Yeah.
On a graph, how do you tell the difference between a neutrophile and
So acetotolerance usually will, a neutrophil usually peaks at 7. So usually we'll see a peak, and some folks in their unknown saw more of like a plateau around 7 where it was kind of like this. Acetotolerant organisms will probably have something more like this first one, but a longer extended range out towards the more acidic pH values. Yeah, that would be the largest difference between the two of them. All right, so for the last set, be able to, or I want you to know the targets of those antibiotics we talked about, what they inhibit. We'll spend more time on spectrum later on. That is something that I do want us to come back to, and we'll spend a couple of days more talking about antibiotics after this exam, a couple of weeks down the road. But I want you to be able to justify using, for example, therapeutic indices as a measurement to determine which of 2 antibiotics is the best to use. We'll take a look at that in a couple of minutes. Analyze results for antibiotic effectiveness. Again, broth dilution, disc diffusion assays as ways to measure how effective an antibiotic is against a microorganism. And then we'll finish up today with talking about methods of antibiotic resistance. Okay. That last one on there, social practices that lead to antibiotic resistance, we will save that for about 2 to 3 weeks down the road. We'll come back to that concept because I think that's a really important one for us to follow up with. All right, so any questions on what's on there? It may seem like a lot, but one of the things that I want to point out is there's a lot of overlap. So for example, what we talked about within the first set talking about growth of microorganisms, the inhibition of growth is based on how do they grow in the first place. And the techniques that we use to determine effectiveness of chemicals is the same set of techniques that we use to determine how effective are antibiotics. So there's a lot of overlap in here. I just want to kind of point that out. All right. So let's move out of there and let's move into something that we began with the other day. I want to go back to this this sheet that we were working on the other day. And when we were working on this, we focused on zones of inhibition primarily. And now I want to finish up our conversation on broth dilution assays and talk about one more measurement that you can make from this, which is known as the MBC, or the minimal bactericidal concentration. So let's spend a little time talking about how do you actually analyze that. Given this problem up here, one of the things that we said was, I left these blank at the time, but if these are twofold dilutions of the antibiotic, the furthest right would be 4 micrograms per mil, 2, 1.5 microgram per mil. The 0 here is just a control to show you that, yes, indeed, the microbe grows, and so any effects we observe are from the antibiotic itself. So given that basic premise, what is the minimal, the antibiotic X, what is the minimal concentration of it, or minimal inhibitory concentration of it with this particular microorganism? 4. And for antibiotic Y? Okay, 2. So, and again, that's just the minimal concentration needed to fully inhibit the growth of the organism. What this doesn't tell us, though, is whether the organism is dead or whether it's just static, whether it's inhibited in its actual growth. And so that's where we can follow up with what we call a secondary experiment known as a minimal bactericidal concentration experiment. So in this one, what we would end up doing, that is definitely not the color of the marker. Okay. So I want to take just one of these as an example. We'll take antibiotic Y. And here we'll represent our growth. Concentration of the antibiotic on top here. And to perform a minimal bactericidal concentration experiment, what we do is we subculture these cells. In other words, we take 10 microliter samples, roughly, from this one and this one. And then we culture them in fresh broth that contains no antibiotic whatsoever. In other words, it's not a selective environment. So we take those bacteria, transfer them to test tubes that have a certain amount of media in them already, and then let them culture overnight. If the organism was inhibited in its growth, but still the cells are alive, what do we expect to observe after an overnight incubation? Growth. Yeah, should be some cells. What if the antibiotic is at the minimal bactericidal concentration? Will we observe growth? No, the cells won't be, there won't be any around to even have growth occur. So the minimal bactericidal concentration is the minimal concentration that not only inhibits growth but kills the microorganisms after 24 hours. And again, the way that we determine this is by performing these, taking a small amount from the original culture, putting it into fresh culture media with no antibiotic, and it's just a binary result at the end. Yes, they grew. No, they didn't. Lowest concentration that stops growth is our minimal bactericidal concentration. Okay? Any questions on that concept? Let's look at an example real quick. All right, so sorry about the crudiness. Can you guys read that somewhat? Sort of. So basically, this is saying growth is on this one. This is growth in a subculture. We have 200, 150, or 25 micrograms per mil of the antibiotic. These are minuses. These are plus to indicate no growth or growth. So based on this chart, I'll give you a minute, identify the MIC, identify the minimal bactericidal concentration for this antibiotic.
What's the difference between MIC and
So the I stands for inhibitory, just saying that it inhibits the growth. It's static, in other words. The MBC is bactericidal, so not only does it inhibit, but it kills the bacteria as well. And you can't tell that just from these experiments, so that's why we have to subculture them to determine the MBC. All right, so given this problem, here's our original culture. Our MIC is what? 100 micrograms per ml, MBC, 200. Okay, the MBC is always going to be equivalent or greater than the MIC, just to keep you, or just so that you're aware. All right, so that's how we analyze that. These results sometimes are used if the goal is to actually kill the microorganisms. But more often we worry about the MIC when it comes to talking about using antibiotics for treatment. Yeah.
how do you know which one is the best antibiotic?
Yeah, let's go over that. I wanted to go back to that problem now. So between these two and the problem that we were just working on, how do we determine which of the two is the best? Okay, and there's a couple of ways we could look at it. One, which one has the lower therapeutic dose? And let's just say in this case the therapeutic dose is equivalent to the minimal inhibitory concentration. So you could argue Y is the best of the two to use. What else do we have to keep in mind though when we think about this? the toxic dose as well. So in practice, we have to keep in mind what amount of the drug causes, at what concentration does it become toxic to a human being as well. So I've written toxic doses there just in that's supposed to be microgram per mil concentration. So what I'll do is I'll give you guys another minute or two to go ahead, calculate the therapeutic index using the toxic dose and using the therapeutic dose or the MIC to determine which of these is a better antibiotic to use on a human being. Toxic dose. What's that? You tell me. All right, so what is the therapeutic index for antibiotic X? 0.5, right? It's 2 divided by 4. And then what is it for the second one? Yeah, 0.125. And again, the equation here is the therapeutic index is equal to the toxic dose divided by the therapeutic dose. In this case, we're saying it's the same thing as the minimal inhibitory concentration. No, not always. And why I say that is because in reality, how these therapeutic doses and toxic doses get measured is through phase or clinical trials, in which usually these oftentimes are provided in concentrations of like milligram per kilogram of the individual, rather than just a set number. Yeah. And we want a higher therapeutic index. So in thinking about how does this relate between the two of them, which one would we choose? X, because not only is its therapeutic index higher, but what does that actually mean then? Well, what it means is that at the dose at which it's being prescribed, it would have fewer, or the severity of those toxic side effects would be less than it is for antibiotic Y. You could argue either. You could, but in principle, when we're talking about human beings, we're oftentimes keeping in mind too the therapeutic index. So if you were to argue, let's say this was like a 3-point problem. If you were to argue Y is better because it has a lower toxic dose, I'd probably give you 1.5 or 2 points out of 3, because you're using the evidence to support your answer. And yeah, lower concentration is probably better to do. When it comes to practice, though, I guess I don't have to preach to the choir, in which most of the students in here are going into nursing. You have to actually care about the patient a little bit as well. So like a lower toxic dose would be better prognosis for life. Another reason why I work not with humans, but only with microbes. I don't have to care about the human being as much. All right, any questions with that? Allie.
So if the therapeutic index is higher, does
It means that the ratio of the toxicity to how much you have to provide to cure the infection is quite large, and then in other words, you can think of that in two ways. One, either the toxicity is really low, or secondly, the amount that you have to, the drug is incredibly effective. And so the amount at which you have to take it is much less than what would cause somebody to become sick from the drug itself. So it's either one of those two things, most likely, but that's why we look at the ratio of the two. It's kind of a measurement of how toxic is it, and how effective is it. Okay, let us then move into the last topic here, and that is going back to talking about antibiotic resistance. And again, when it comes to antibiotic resistance, we will come back to this, but what I'm going to introduce you to today is just a couple of examples. There are 4 major ways in which we can consider antibiotic resistance to develop, and so I'll go ahead and cover those. So, in general, organisms can be naturally resistant to antibiotics, or they can somehow gain a resistance. And oftentimes when we say that organism gains a resistance, what we mean is that they have spontaneously gained resistance through a mutation to the DNA, and so it's changed the structure of a protein so that the antibiotic can no longer bind to that protein as well. Or the bacteria has actually gained a full new gene through a process known as horizontal gene transfer. And that's a topic we'll come back to later. And we'll come back to mutation as well and take a look at some specific examples for that later too. So I don't want to spend much time talking about those today. Instead, I want to spend a bit of time talking about the other methods by which antibiotic resistance can occur. And these come down to functions of specific enzymes or characteristics of the microorganisms. So we've talked about, for example, penicillin is not effective against gram-negatives. It's only effective against gram-positives. What is one potential explanation for that? Yeah, the outer membrane. That is a natural structural barrier that exists in gram-negatives and actually makes them resistant to quite a few antibiotics. In the same sense, some of those mycobacterium or those acid-fast bacteria that have the mycolic acid on the outer wall, outside that outer wall, they are resistant to a number of antibiotics as well because, well, that mycolic acid is waxy. Things can't permeate through that, or antibiotics can't permeate through that in the same way that they might be able to through a pork protein, for example. So there's some natural barriers that exist already. Here are a couple of other examples of barriers that exist. Some microorganisms, and this is one we'll talk about today, have enzymes that will inactivate the antibiotic, actually lead to the degradation of the antibiotic. Classic example of this is the hydrolysis of penicillin by a class of proteins known as beta-lactamases. We'll come back to that. The other is through not necessarily cleaving or cutting the antibiotic, but adding something to it that inactivates it. So chloramphenicol, which targets the ribosome, can be modified with an acetyl group, and that the enzyme that drives that reaction is known as chloramphenicol acetyltransferase. Not important. But the important part is once that acetyl group gets attached to it, chloramphenicol is now inactive. Can be floating around the cell, but it does nothing to inhibit translation anymore. So those are just two mechanisms where the effect is directly on the antibiotic itself. The last one over here, target modification, this is mutation. We'll come back to this. So changing the structure of the protein in some way so that it now no longer can be recognized by the antibiotic. But the one on the right or the left over there, this membrane modification, this can be things like we've already said for gram-negatives, they have the outer membrane. That protects them against antibiotics. Some organisms might undergo further modification in which the structure of the phospholipids, or the type of phospholipids in that outer membrane or inner membrane, make the membrane less permeable to antibiotics. One thing that Pseudomonas aeruginosa does, and some other bacteria have this as well, is they've got outer membrane proteins. And we've described some of those outer membrane proteins as being porins, or pore proteins that will allow for molecules to come in or come out of the cell. Well, the pore protein in Pseudomonas aeruginosa, it's known as OPRM. And we're going to talk about this one probably in about 2 weeks from now, because there's some really interesting studies going on with it right now. But these proteins are what we call efflux pumps. As soon as the antibiotic makes its way into the cell, the organism with these efflux pumps uses an active form of transport to basically force that antibiotic right back out. So it never comes into contact with the actual target that it's intended to work upon. So I'm going to give you an example of what does this look like, not so much in practice, but what do the results from this look like? So here is an experiment that was performed, 22 years ago. So this experiment is older than some of you, if not many of you. And in this particular experiment, what the researchers were doing was they were testing two strains of Pseudomonas aeruginosa. So the K372 there is what they're calling the wild type strain of Pseudomonas. And then they actually went into the genome and they deleted this efflux pump protein so that the cells no longer could express it. And then they tested how sensitive or resistant are these wild type cells and these mutants to the antibiotics that they were testing against. So they're testing norfloxacin, ciprofloxacin. These are both quinolones. What is the quinolone target? Replication, gyrase. Tetracycline and chloramphenicol. What's chloramphenical's target? And then novobiocin. And what I want you to do is just look at those 5. Take a look at the minimal inhibitory concentrations in the wild type, and take a look at the minimal inhibitory concentrations in this strain here, which is where they knocked out that efflux pump protein. Okay? Take a second or a minute to look at the two, and then just describe what is the general trend that you observe. All right. So generally, what happens to the minimal inhibitory concentration in comparison between the wild type and these cells lacking OPRM? The numbers are smaller. In other words, are these cells more sensitive or more resistant to the antibiotic? Yeah, they're more sensitive. It takes less antibiotic to inhibit their growth. And so what they were concluding from the study then was that this protein is a major contributor to antibiotic resistance for Pseudomonas aeruginosa. And what some groups have done since this time, and one of the papers we'll look at down the road, is they've tried to target this protein specifically. Can you block it in some way so that these cells no longer can pump out the drug? And some of those strategies are being used in the medical field now. So we'll take a look at that later on. It's a really interesting paper. But let me show you the inverse of this real quick. They also then tested 2 other strains, so this PAO strain and then PAO, in which they put in OPRM. They started overexpressing it. So put it in a way that it's expressed all the time in these cells at a much higher concentration than you would normally find. And what happens there when you overexpress this gene? What happens to the MEC? So we'll just describe the trends for each of these. So for that first antibiotic again, which was norfloxacin, I think, 0.5 to 2 increase. The next one was Cipro, 0.1 to 0.8 increase. And we find that again and again. So what this is showing is that if the cells are engineered in a way to overexpress this protein, or if there were some mutation in the genome that allows for this protein to be constantly expressed at very high levels, the cells actually become more resistant to antibiotics. And that's what they're trying to show here. So this method of efflux pump is an important one that probably hasn't gained as much attention as it probably should, though there's more attention being paid to it now. So questions on this mechanism, and I'm just using this example here to kind of give you an idea of the ways that resistance can occur. Yeah.
For the antibiotic modification, should we just know that it's like adding something as a modifier?
Yeah, that's the main idea. That is the main idea, is that it changes the identity of the antibiotic in some way so that it no longer can target the protein it's intended to target. What about it? Know it as a mechanism for right now. We will come back to this later. We're going to kind of do a whole second pass of antibiotic resistance, but in the context less of like the measurements and more of like at the cell level what's happening. Now, I'm going to go over one more, and this drove people nuts this morning, so I apologize. Bear with me. But I feel it. I have to do my due diligence to explain this one. All right. The other mechanism, which has gained a lot of attention, and in part because this has led to resistance of many types of microorganisms worldwide, actually, is through what we call beta-lactamases. The beta-lactam, anybody remember what's the beta-lactam structure is? We kind of talked. Yeah, it's the ring. Good. That's a great start. So penicillin, here's that ring structure. What beta-lactamases do is they actually take that ring structure and they cut it. And in doing so, it inactivates penicillin. So taking penicillin, hydrolyzing this bond right here between this carbon and nitrogen leads to an inactive form of the antibiotic. At this point, it's useless. No good to us because it no longer can inhibit cell wall synthesis. So what we find in some microorganisms is that they encode these beta-lactamase genes, and the abbreviation is BLA, or BLA, is the abbreviation for that. And I'll try to draw out how this works, essentially. So if this is our beta-lactamase, blah, and this is penicillin, penicillin fits into the active site of beta-lactamases. It also fits into the active site of penicillin binding proteins, as we've already talked about a couple of days ago. And so there's our penicillin. I'll label it with a P for now. Penicillin binding protein here. We described that when penicillin fits into the binding site of penicillin binding protein, it blocks cell wall synthesis. It functions as a competitive inhibitor. Okay. Well, penicillin's a substrate for beta-lactamase. And so when it fits into the beta-lactamase active site, beta-lactamase cuts it, and what comes out here is an inactive form of penicillin that no longer functions. In other words, it loses the shape that's required for it to function as a competitive inhibitor. And so it no longer fits into the active site, of the penicillin binding protein. Everybody following so far? So basically, the goal of the beta lactamase is to inhibit an inhibitor, in that sense. However, we have found ways to overcome this effect of the beta lactamase. It's by using one more class of antibiotics, the last one I will ask you to know about, which are the beta lactamase inhibitors. And these beta lactamase inhibitors look a lot like beta lactams to begin with. So on the left over here is an antibiotic known as clavulanic acid. On the right is amoxicillin, which classical beta-lactam structure. Clavulanic acid has it as well, smaller molecule though. Together, these two are delivered in the form of augmentin. So if anybody's had augmentin or a derivative of that, it's these two drugs combined. Okay. So this is how they function. Clavulanic acid fits really nicely into the active site of beta-lactamase. Doesn't do as good of a job fitting into the active site of penicillin binding proteins though. And so in essence, clavulanic acid is a competitive inhibitor of the beta-lactamase. I need another color. So here's that clavulanic acid. And what happens when clavulanic acid fits there? Can beta-lactamase now cleave or hydrolyze the beta-lactam antibiotics? And the answer is no. It can't function in that way anymore. So in other words, clavulanic acid is an inhibitor of an inhibitor of an inhibitor. Phrased another way, it blocks the activity of beta-lactamase. So what we have here is a two-hit system in which clavulanic acid and amoxicillin work in synergy with one another. Clavulanic acid blocks the beta-lactamase from functioning, and that then increases, in some ways, the active dose of penicillin and allows for more penicillin to be present to block cell wall synthesis. So how would this look then if we were to analyze this in some data? Let's take a look at that real quick. Well, let's just start with a simple minimal inhibitory concentration experiment. Okay, I have two things I want you to do. One, given this problem right here, identify what the minimal inhibitory concentration of amoxicillin is. That's part one. And then two, assuming this bacterial strain that we're using in this experiment, let's say it's E. coli, assuming that it encodes a beta-lactamase, what would the results look like when we have amoxicillin and clavulanic acid at the same time? Would these cells be more sensitive, or would they be more resistant to the combination of those two drugs? So I'll give you a minute to draw that out. Draw it out first. Yeah. So some cells encode this beta-lactamase, not all bacterial strains do this. If they have this beta-lactamase, they can So the CA is the coagulonic acid. It's a
Yeah.
so yeah, you've got CA. It basically causes So after you find the MIC for amoxicillin, given let's assume that these cells have a beta-lactamase inhibitor. What would draw an example of what the MIC or what the results might look like down here if these cells were treated with augmentin, so clavulanic acid and amoxicillin at the same time? Augmentin is the combination of the two of them.
Is there a reason it's zero?
Just because that's what I wanted to do.
I've been taught that it usually like plays out.
I just drew it a different way. It's translatable. It's translatable information. Some people do it right to left, others do it left to right. Some do it top to bottom, some do it bottom to top.
Yeah.
But there is. It's a decreasing concentration from 4 to 0.25. Yes, it's consistent. Because that's what I wanted to do. Here, I'll do this. To appease everyone, now we have double the controls. All right, what's the MIC for amoxicillin? 4. Good. What might be the MIC for augmentin, the combination of these two drugs? 0.25, 2, anything less than 4, right? So if these cells have a beta-lactamase, what's happening here? Well, they're cleaving the amoxicillin, which means that compared to a cell that doesn't have the beta-lactamase, this MIC is greater. And now if we inhibit that beta-lactamase with clavulanic acid, in some way it decreases that MIC, or that minimal inhibitory concentration. And that's how these two drugs work together. Okay? So, I've already revised the exam because this actually was the original last essay question, and I went, oh shit, we're in trouble. So, I sometimes fail how complex of a concept this really is. There is a question on this. It's not an essay question, though. It's a multiple choice question. Okay? So, I love you guys enough to do that. Yeah.
So the right answer would be anything less than 4.
It'd be anything less than 4. Yeah. So.
We have to, is that like all that we would ever need to just
So for a problem like this, you might have to say, like, given these results, do you think the organism has clavulanic acid? Or I'm sorry, do you think the organism has a beta-lactamase? If you saw something like this, where the augmentin had the same exact effect, you might conclude that no, there's not a beta-lactamase, because if there was, well, then the clavulanic acid should have inhibited it in some ways, and that's not being seen here. So you might make an assumption that it's a different form of resistance to amoxicillin, an efflux pump, a mutation to a penicillin binding protein, for example. All right, any last minute questions? Just wanted to humble everybody one last time before we go into this next exam. All right, good luck. Have a nice weekend. Try to get some relaxation time. I've got a short meeting right now, and then I'll be around. But I will be around till 4.
We check your emails.
Yeah, occasionally.
Yes. What's the best time to email? 10 p.m.
I'll probably respond like tomorrow morning and Sunday morning. Definitely are times that I'll be responding. Do not contact me at 10 o'clock at night on Sunday expecting a response until Monday morning. She's not going to be able to. She's got apparently a really busy week coming up. So I did ask though.